Thank you for joining us for another presentation on the aspects of method validation. Today we'll be going over precision. Precision is fairly simple to execute, so we will quickly go over what precision is and the three parts that are required by the ICH guidelines, and then discuss when it should be performed. Precision is the ability of the method to get the same result many times and has a component of accuracy embedded in it. But it differs from accuracy in the results measured. For precision, the results of multiple replicates must be similar to each other regardless of the value of the results. For example, 6 preparations of a drug product sample yields results from 88 to 90% of label claim. This shows precision, but the results may not be accurate since the results are outside of the general accepted range of 90 to 110%. There are 3 main parts of precision: injection repeatability, sample preparation, and intermediate precision. We will discuss each in more detail, starting with injection repeatability. Typically, a single preparation of a standard or sample is injected 6 to 10 times. A requirement for the %RSD for peak area of the peak of interest is set to less than 2 for HPLC and less than 5 or 10 for GC methods. Most of the time, the standard preparation is used for this assessment because injection repeatability is assessed as part of system suitability. Next up is sample preparation precision. This is performed by preparing 6 independent sample preparations per the method. It's important that the same lot of material is used. This should be done for API and DP samples. For methods such as content uniformity, 10 sample preparations are performed because that is what is required for the test as a whole. This criteria is based on the type of assay being performed and the expected variability of the product being tested. For API, a %RSD of less than or equal to 2 is common, while a DP method may also have a %RSD requirement of 5 or 10%. For impurities methods, precision also needs to be demonstrated. Once again, the criteria is dependent on how the impurities are calculated, either %area or %weight. For early phase programs, a %RSD requirement may be placed on the total impurities rather than the individual impurities. Intermediate precision really tests the precision of the method by introducing several variables into the evaluation. It requires at least 2 analysts performing 6 sample preparations per the method. Each analyst should prepare their own mobile phases, diluent, dissolution media, etc. If possible, different lots of columns should be used as well as different instruments. A requirement is set for a %RSD for all 12 results. Sometimes an acceptance criteria is set for the agreement of the results. As shown, precision is required for all quantitative assays at all phases of the product life cycle. Intermediate precision is not required for phase 1 programs. Abbreviated intermediate precision is performed at the phase 2 stage where 2 analysts perform these 6 preps but have no additional variations introduced into the study. Phase 3 programs must have the full intermediate precision evaluation done. It is important to consider the method and how the variations that are included in the evaluation may affect the results. We had an issue where a different sonicator was used by the second analyst. That sonicator produced more heat and caused the impurity results to be significantly different than the other analyst. So think about all the steps and what's important to be evaluated when developing your protocol. This concludes our discussion on precision. Please reach out if you have any questions or comments. Thanks so much for watching.